All right. Hey, everybody. My name is Elliot Kimmel. I'm a high school science teacher at Central Secondary School. And this is my tutorial on mitosis, cell division, cell cycle, that kind of stuff. So in order to get the best out of this video tutorial, I want you to have some diagrams of mitosis. So if you're using the Nelson Science Perspectives 10 textbook, then that's ideal. You can look at the pictures on cell cycle and mitosis that are in there, and I'm going to be drawing some of those from that textbook. We're going to be using those here in the video tutorial. If you're not at Central, you're just some student somewhere else studying mitosis, hopefully you'll still find this helpful. Okay, this is brought to you by www.zerobio.com, which is my science website. Lots of resources there for you for various grades. Okay, so check it out sometime. Okay, so let's start right over here with the cell cycle. So you should have this diagram. If you're in my class, you'll have this diagram on a printout, the cell, which is called the cell cycle. So I'd like you to have a look at that right now. And if you're not finished labeling that diagram, then add some of these labels as we go along. And anything in particular that I write, you should also add, and I'll make special note of that once in a while. So this is the cell cycle, and what it basically is an overview of the life cycle of a cell, all right, from birth through growing all the way to dividing and reproducing. All right, so we got to choose a place to begin. So let's begin right here where it says the cycle begins. Now, what they draw here is they draw a cell, and this purple area, I guess, sort of represents the nucleus. And inside here, you've got one chromosome. So that's a chromosome right there. And they don't make a distinction here about chromatin or chromatid or anything like that. We will do that soon. So if there was only one chromosome in a cell, and I can't think of any species that only has one, but assume if there was, at some point this one chromosome is going to have to be replicated, as you can see down here, DNA replication, all right? And you can see an X structure here, which is really two chromosomes that are joined. Now, just give me a minute here, and let's actually talk about why cells are going to reproduce. I'm going to get rid of those labels. Come back over here, and imagine that you've got a cell, and I'm not going to draw a circle. I'm going to look something like a cheek cell, right? And there's the nucleus in there, and let's assume there is one chromosome. Now, at some point in time, this cell is going to divide or replicate, and it's going to use the process called mitosis to do that. So here we have another cell over here. This is going to be a daughter cell, and another cell over here. This is going to be another daughter cell, all right? And each of those has to inherit that one chromosome. So I'm going to draw a chromosome in here, draw a chromosome here. Now, you don't have to be drawing this diagram. If you do, there's no problem. This is the process called mitosis, a summary of mitosis right here. And in order to get two daughter cells, each with the correct number of chromosomes, at some point along, this cell is going to have to replicate its chromosome or its DNA, and it will look something like this. All right, it'll be joined together at a little spot there. So it's going to go from one chromosome to two, and then it's going to split, and each of the daughter cells is going to get one of those chromosomes. All right, so this DNA replication has to occur before the cell divides. So this occurs during a part of the life cycle, which is called interphase. And during interphase, the cell is doing all the things that it does normally in life. So it's taking in nutrients like oxygen and glucose and other things, and it's getting rid of wastes like CO2, carbon dioxide, and maybe a little bit of water is coming out, and a little bit of water is going in as well, and all that kind of stuff. And it's growing. And then at some point, it's going to replicate those chromosomes so that it can then go through the 4 stages of mitosis to create daughter cells, each with that one chromosome. These daughter cells are going to be genetically identical to the parent, all right? And that's simply because that chromosome is just replicated. It's copied like a photocopy, all right? So there's a little bit of background about mitosis. So let's go back now and look at that cell cycle here. And of course, I've lost my original labels. We don't need those anymore. So we're starting the cycle here, and as you can see, the cell grows. It will do various things throughout its life cycle, but at one point down here, before mitosis, it will duplicate that chromosome or its genetic material. And that's what we see here. Then the cell will prepare to divide, and only when we get up here at like 11 o'clock on the clock thing here, okay, for 1 hour between 11 and 12, let's say, that's when mitosis will occur. And as you can see, cell division occurs, and we're going to create two cells, and we're going to split them. And the splitting of the cell is called cytokinesis. It's not a phase of mitosis. Mitosis involves the nucleus and the chromosomes. Cytosis refers to the splitting of the cytoplasm, kind of like what you're seeing here with the splitting there. And then we'll have our daughter cell there going off with the same number and type of chromosomes as the parent. So this is the overall cell cycle, and mitosis is just a small part of that cycle. Now, if we come over here, let's use another color, we see another image. This is from the Nelson Science Perspectives textbook, all right? We see another picture where DNA is being copied or replicated. Now, when we say DNA, that's this stuff. The chromosome is DNA. It's supposed to be an arrow there. All right, so what you see here is one DNA, and here you see the word DNA written down twice, and that implies that the DNA or chromosomes have been copied. Then mitosis occurs, and that cell goes through the division process, and then cytokinesis, which splits them in half, and that is the process of cell division to create two daughter cells by cell division, and both of these will have one DNA in this case, the same as the parent had. All right, so there's a little overview of interphase, mitosis, and then cytokinesis. So let's get into the details. This diagram is the one that I asked you to either draw or to photocopy, and it represents prophase, which is the first step of mitosis. Now, you want to make sure that you have all of these labels here, and I'm probably going to be adding a few more as well. What you can see in prophase, which is the first step of mitosis, this comes after the cell has gone through interphase. So the chromosomes or DNA have already replicated. So if we look at this green one here, we see a worm-like thing there and another one there. Both of these are chromosomes, but at this point, when they are joined in the middle, this structure here, I'm going to darken this one in here. This is labeled as a centromere. This is a structure that holds together the two replicated chromosomes, and because they are identical to each other, they're copies, these are called sister, and I'm going to get you to label this as well, sister chromatids. Now, there's a label over here, chromatids, so it's pointing at one of them. There are two sister chromatids, each joined by a centromere. So that's a sister chromatid, that's a sister chromatid, another one here, another one here, another one here, and another one here. All right, I'm just going to clean that up so it's easy to see, as long as you label the sister chromatids. Okay, let's come back here. Now, you're going to notice this label right up here, chromosome, with a bracket. What they're saying is this whole structure can be considered as a chromosome. Now, you might say, well, you know, is it two chromosomes because it's replicated, it's doubled? Well, it is replicated and doubled, but because they're identical to each other, they're just copies. And you'll learn how that happens later on, most likely in grade 11 biology. But because they're identical to each other, it's the same thing. It's the same chromosome. It's kind of like when I give you a handout in class, all right? There's 30 of you. I give the same handout. It's one handout, but I've got 30 copies of it, all right? So this is a chromosome made up of two chromatids joined together by a centromere. This is DNA. So if you were to zoom in on it really close, you would see that double helix structure of DNA, something like that, right? That's what it would really look like deep down inside. Now, you're also going to notice in prophase this purple area. This is the nuclear membrane. So if you could add that to your diagram, if you've got a diagram there, or if you're drawing these, And it looks kind of blotchy here because the nuclear membrane is dissolving. It is breaking down. And the reason that that is going to happen, and this is the area where the chromosomes are, the reason the nuclear membrane is going to go away is because out here in the cytoplasm are some structures, and I want to draw these on because for some reason they're not illustrated. And I'm obviously not a great drawer, as you can tell by this presentation. I'm just going to draw a circle there, and I'm going to draw another circle over here on this side, and I'm going to label this centriole. Now, this probably represents an animal cell going through cell division. It's a little bit different in a plant cell, but the centriole creates these black structures that come sticking out, and I just want to find a good spot to label it. I think I'll label it over here. I'm going to grab one of these, so add this label. This is called a spindle fiber. So the centrioles, and there's a pair right here. There's really two of them in there, so we could do something like that if we wanted. The centrioles on either side create spindle fibers, which radiate out and will become attached to the centromere of each replicated chromosome, as you can see over here. We'll talk about that one in a minute. So during prophase, the spindle fibers are being created by centrioles. In order for the spindle fibers to actually touch the chromosomes, which are inside the nucleus, we have to get rid of the nuclear membrane. So the nuclear membrane here is going to break down and disappear so that by metaphase, there's no more nuclear membrane and the spindle fibers can touch the centrioles. So in prophase, we see the replicated chromosomes made of two sister chromatids joined at the centromere. The nuclear membrane is dissolving or breaking down. The centrioles have moved to opposite poles, whether it's on the left or the right, or the top and the bottom, whatever, and spindle fibers are forming. So let's move on to step 2 of mitosis. This is called metaphase, as if chromosomes have met in the middle. So here we can see they are lining up along sort of an imaginary equator of the cell. We can see the sister chromatids right here. So once again, these are sister chromatids. The sister chromatids are joined in the middle at the centromere, and they are lined up along the middle. Now, by the way, if we count how many chromosomes there is one chromosome made of two sister chromatids. Remember, they're identical. 2, 3. Now, this is not the number of chromosomes in a human cell, all right? This is just for simplicity. They're drawing 3. There would really be 46 of these things in humans. 46. We have 46 chromosomes in our human cells. All right, but that's too many for this diagram. Now, here is where the centrioles would be again. And you can see little black things coming out like a little starburst. So these are little baby spindle fibers, and these ones are a little bit more mature. So let's label this as a spindle fiber. And the spindle fibers have oriented the chromosomes at the equator of the cell. Maybe I can even label that. Bring a label down here. Call this the equator. I know this looks messy. Hopefully your notes are going to look a little bit better than mine do. All right. That's metaphase. All right. And we're ready to move on. I don't think there's anything I've forgotten. We'll see. All right. So I'm going to clear this up. Make sure that you've got all those labels down. I'm going to clear this up. And then we're going to move on to the second half of mitosis. All right, here we are in anaphase. The centriole still would be in there. There we go, centrioles. And now what is happening is the spindle fibers, these black lines, are pulling towards the pole. So I'm going to draw little arrows. I suggest that you do the same. These arrows represent the direction that the spindle fibers are pulling. They're pulling the chromosomes back apart. Now, I just said the word chromosome there. I mean, I said the word chromosome a few times. I've also been using the word chromatid. At this point, when these are separated from each other, they're no longer called chromatids or sister chromatids. They are now called daughter chromosomes. That's daughter chromosome. That's one daughter chromosome. There's another daughter chromosome here, 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 and here. All right. And as you can see, 3 daughter chromosomes in this diagram are being pulled to each pole by the spindle fibers. And this is anaphase, where the chromosomes are now separated and they're moving to the poles. In telophase, the chromosomes have been pulled to opposite sides, the same number of chromosomes. Remember, this one here, this red one, is identical to this red one. They were copies. The blue is identical to the blue, the green to the green. So the same number and types of chromosomes are pulled apart. And now we can see that a nuclear membrane is reforming around each group of chromosomes. So let's label nuclear membrane. The nuclear membrane is reforming here around each group of chromosomes. The spindle fibers have disappeared or are in the process of disappearing. All right. The centrioles still exist, but they're not drawn here. They're not critical. They're not performing the role anymore. And we're just getting ready, because what's going to happen now, this is the last step of mitosis. The next thing that's going to happen is the cytoplasm is going to pinch in like this, a little bit at a time on both sides. And then it's going to, these pieces are going to join. It's going to be a little bit more, and eventually they're going to join, and the cell is going to split down the middle. That splitting is called cytokinesis, and it occurs after telophase, so it's not part of mitosis. Mitosis involves the chromosomes and nuclear events. Cytokinesis occurs after, and it splits the cytoplasm, and we will end up with 2 daughter cells, one going that way, one going that way. These will be our daughter cells right there, 2 of them, and they will be identical, and mitosis has done its job. Okay, let's have a look at mitosis for real, and some real images, and see what we can identify here. Now, this is a microscope slide similar to something that you are going to see. This is an onion root. Now, there's nothing for you to draw at this point. I just want you to pay attention and get this, because your challenge is going to be to identify these stages very soon for real. Let's see what we can identify. Now, most of the cells in this slide are in interphase. They're in the, well, it's called a resting phase, but they're not really resting, all right? They're growing, they're metabolizing, they're doing all those characteristics of life type things. So, for example, this cell here, what we can see is a nuclear membrane right there, and you can see a nucleolus, that's that dot right there, all right? So this cell is in interphase. You can't even really see the genetic material very clearly like you can see it here, these black worm-like things. Here's another cell that looks like it's in interphase, and this one, maybe not this one, but this one, and this one, and this one, and this one. So over here where there's the label C, these cells are going to be in interphase. At some point, the nuclear membrane is going to start breaking down, as it looks like it is here. And you're going to start to be able to see the chromosomes just a little bit. This would represent prophase. So over here in D, this looks like a little bit later prophase, where you can see the nuclear membrane is kind of disappearing. You start to see these black lines in no particular arrangement. That would represent prophase. Okay? I mean, it's difficult to do, but that's how it's done. After prophase, we have metaphase. And these are some beautiful examples of metaphase right here. So you can see these black worms roughly in the middle of the cell. Now, don't get confused. Don't think that they're always going to be perfectly lined up in the middle, because think about it. This is a dynamic process. And I'll give you an example. If I were to ask you how old you are, all right, how old would you say you are right now? Would you say you're 13 years old? Or would you say you're 14 or 15? How many of you would say, well, I'm 13.4 years old, right? You wouldn't. You would probably take an average or a generalization. And that's the idea about mitosis as well. We go prophase, metaphase, anaphase, telophase, but it's not like it clicks from one frame to the next. It's prophase, and all of a sudden it's in metaphase. So when you see an image like this, all right, you have to figure out, well, what phase is it closest to, or what is it, in general, what phase is it in, and it's moving out of the previous phase and into the next phase. So in metaphase, the chromosomes are lined up in the middle, but they might flop over this way and that way, and they're getting ready to be pulled to the poles. So there's a beautiful example of metaphase. This one over here also looks to some extent like metaphase. The odd chromosome is looking a little bit more floppy, and in one direction or the other. All right. Now, there's E. If we come over to this one here, this one that's labeled B, you can see the chromosomes are sort of, I almost see the fact that they're being pulled apart. I almost feel like I see something in the middle here, like spindle fibers. So this would represent anaphase, and then this would represent telophase. Now, this is a plant cell, not an animal cell, all right? So it doesn't exactly undergo cytokinesis to separate them. Instead, a cell plate is put down here, a line, and then a cell wall develops, and the two are separated, but they're like bricks, because this, like I said, is part of a plant. This is the onion root, all right? If this is an onion, okay, these little roots grow out of it, or think of any roots in any plants, whatever. That's my impression of roots. And the cells stay together like a brickwork, so they don't completely separate. But you don't necessarily see the development of a cell of a nuclear membrane right now. And that's fine. That doesn't mean that it's not telophase. The chromosomes are over in one bunch over here, and I just draw the nuclear membranes there and say, okay, there, did I convince you? That's telophase. All right. If you want to see a better telophase, come on over here down to this picture. And again, this is a plant cell. Yes, they're rectangular. It's not a great way of identifying them, but you can see all the chromosomes sort of over here, and the little hint of a line developing here. This is going to be called the cell plate, all right? And then you're going to have two individual cells, all right? So that's going to be telophase, and this is a picture of an animal cell dividing, and you can see all the chromosomes in a bunch right here, and yes, the nuclear membranes will form at some point, and it's pinching in here, and it'll pinch in right there. So that's evidence of cytokinesis. So this is telophase going into cytokinesis, and then you will have two animal cells right there. Stuff, though, isn't it? Let's have another look at something else. Okay, now, these are not my microscope diagrams, so I can't take any credit for them. These are from the internet, but they work well in this slideshow. Can you recognize some cells that are in interphase? All right, how about this one right here? Does that look like maybe it's an interphase? This, by the way, that's the nucleolus. All right, this is where ribosomes are produced. That is the nuclear membrane. That is the cytoplasm. And there we see the outside of the cell wall or cell membrane. All right, so look at this one, and this one, and this one, and this one, and this one. Now, what about this? Looks like a little ghost. I'll put a little happy face there, right? Why are there two dots here? This is not the DNA, okay? This is the DNA. These are the chromosomes. There's another one with two, Cells can have more than one nucleolus. A nucleolus is just the region where the ribosomes are being produced. It's not a membrane-bound organelle like the chloroplast or the mitochondrion or a lysosome. It's just a region of dense material. So there can be more than one nucleolus. All right, so don't let that bother you. Most of the cells are in interphase. Now, if I look for a cell that's in prophase, what I'm looking for is a darkening up inside the nucleus as the chromosomes thicken. Now, here's something that I probably didn't tell you about before. I'm going to draw a cell, a square cell here. Pretend it's a plant cell. And I'm going to draw the nucleus and switch to black. And inside the nucleus are the chromosomes, the DNA, right? Well, At some point in the cell's life, the DNA is a long, stringy thing, very thin, and it's called chromatin. Some people say chromatin. Okay, chromatin, whatever. Long and thin. When the cell is getting ready to divide and go through mitosis, draw another cell here, the DNA, the chromatin thickens up like this, and it becomes much more visible. And the cell is getting ready to go through mitosis. Now think of it this way, all right? Here's my analogy. Mitosis is like moving from one house to another house. The DNA is going to move. It's going to move into two houses, two cells. If you were moving from one house to another, you would pack up all your belongings in boxes and bags, throw them in the moving van, right? All together. You can't move when your stuff is all over the place in your room and in your house. It's impossible to do that. So you compress or condense all your belongings so that you can move. And this is what the cell does when it's preparing to divide. It compresses or condenses its genetic material into these bodies that we can see called chromosomes. Chromosome. So we go from chromatin to chromosome, and it's the chromosomes that are thick. And that can tell you, oh, this cell is getting ready to divide. It's going to go through mitosis. So that's what I'm looking for when I look at these cells. Here, I just see very faint fiber thread-like stuff. It's going to be chromatin here. But when I see this, and this, and this in particular, I can tell those are chromosomes, and the cell is actually dividing. So, interphase, prophase. And the reason I say that is because it's getting darker now. I can see the genetic material darkening in there. Now, can I see another prophase? Doing this on the spot really quickly. This is what you got to do. You got to scan. You know, this one kind of looks like it's prophase. I mean, you know, as opposed to that one, which is interphase. Yeah, I'm not sure what to do about this one here. All right. Anyways, maybe you see one that I don't see. All right, let's go on. So we got prophase. Let's look for metaphase. Do you see a metaphase anywhere? Oh, I see a really good one. Do you see that one? Yeah, down here. Okay. Metaphase. These are the thick chromosomes lined up in the middle. What about this one? If push comes to shove, what are you going to call that one? I'm going to call it metaphase. I'm not going to call it anaphase, although it looks like the chromosomes might be starting to be pulled. I like this one for metaphase because I like this one for anaphase. I like this one for anaphase. What else? Nope. We're coming there in a minute. Oh boy. Okay, let's go with that. All right, let's go for telophase now, right? Telophase right there. Chromosomes are in a bunch. They've already moved houses. You guys live here. But by the way, you're identical, right? If there's 46 chromosomes in a human here, there's 46 here. Of course, this is an onion root, so it's not the same number of chromosomes. You can start to see this area here. This is the cell plate. A cell wall is going to be laid down. And all right, we live in two different apartments. I like this one for telophase as well. And that's good enough anyhow. We're not going to see cytokinesis, okay? We're just going to see the cell plate being laid down here because this is a plant cell. So that's how you do that, okay? And you're going to get a chance to look under the microscope. But you know what? Believe me, it's not going to look this big and this clear. It's tough. Okay. What's next? All right. Well, I guess I've said enough. It's time for you to do your assignment. You should have a copy of this worksheet, or you'll get a copy of this worksheet. It looks like this. There are 3 sections to it. 1, 2, 3. What I want you to do is to name the structures that I'm pointing at as best you can. All right. If you're unsure, then do it in pencil. All right, so we can fix it up later. We may even actually add some more labels. I want you to name the phases of the cell cycle, keeping in mind it's not necessarily just mitosis. And by the way, there's the cell plate right there. Okay. Then you come down to this one, and I want you to also name the phases of the cell cycle. Okay. You can color these in if you want, and then I'm going to get you to indicate the letter for the image above up here for each of these little phrases. At what letter and therefore at what phase do each of these occur? Okay. So I hope that helps you understand mitosis. Please visit my science website. Look for some more stuff on mitosis. Let me know if there's anything that you need to hear about, you want me to teach you about, and I'll try to put a video up here on YouTube or make a little animation or a game for you. All right. Hope that helps. Take care.